Hi, I'm Larry Johnson. I am a professor here at Texas A&M University. And today we're going to talk about muscle. We're going to talk about features and functions. We're going to talk about the types of muscle, smooth muscle characteristics, and their locations.
Now, what's the function of muscle and what are some features of muscle? Well, the function of muscle is to generate contractile force. That's the overall thing that it does. Locomotion of multicellular animals, the beating of hearts and movement of internal organs depend upon muscles of different types. And we're going to talk about those types. Distinguishing features of them, muscle has a high concentration of contractile proteins, actin and myosin, and sometimes they're arranged diffusely, like in smooth muscle, and other times they're arranged in sarcomeres. Sarcomeres is the functional unit of stratified muscle. And all three types of muscle, cardiac, skeletal, as well as smooth muscle, all of those come from the mesoderm. So there are three different major types of muscle. You've got skeletal muscle, which is a large cell, and the nuclei are on the periphery, not in the center. It's voluntary. That's the one that we can control. It's large cell, multinucleated, and it has striations, as you can see here. Also striated is cardiac muscle. Cardiac muscle is much smaller. They're branched. The nucleus is in the center. But they do still have striations, and the functional unit is a sarcomere. Branch cell, nucleus in the center, usually one cell and one nucleus, but it could have more than one. Smooth muscle cells are involuntary. We can't control them. And they have a single nucleus, and it's non-striated. The action of myosin is aligned more diffusely instead of in sarcomeres. So if we look at smooth muscle, we see that these are kind of like cigar-shaped cells, and there's a nucleus that is seen. The nucleus is inside the cell itself. So this is a muscle fiber or muscle cell. And if we look at that, we can see individual muscle cells. There's a nucleus of one, there's a nucleus for another one, and you can see the kind of pink cytoplasm. Here we can see cross sections of them, the nucleus in the center, but you can see the individual muscle cells there, and it is non-striated, as you see there. If we look at the ureter, which has lots of smooth muscle in it, we can see bundles of smooth muscle here and here and here and all around through there. And it's a little more dark than is the connective tissue that you see here underneath the epithelium. It's a transitional epithelium since it's from the ureter. And then this is connective tissue below. There's also connective tissue on the outside. So we have fibroblasts, and then we have bundles of collagen that we see there, which is lighter staining than is the smooth muscle that we see here. Smooth muscle, you can see a smooth muscle bundle and individual cells there. If you look at a higher mag, you can see individual cells. Here's a drawing that we saw, and this is the actual cell. These are nuclei, so these are multiple cells. This is an individual cell you see there, and it's non-striated. If you look with toluidine blue, this is a ductus deferens, so this is a pseudostratified columnar epithelium and the lumen in through there, but all this is smooth muscle, and we can see individual muscle fibers or cells. So you can see the nucleus in the center of these, but it's non-striated, as you can see. If we look at rat intestine, we can see the mucosa there, which is epithelium plus the lamina propria, the connective tissue, all the way to the muscularis mucosa. There's muscularis mucosa and a muscularis externa. Both of those are smooth muscle. If we look at those a little closer, smooth muscle are the muscularis mucosa and the muscularis externa, and we can even see the myosin plexus that regulates the contraction of the muscularis mucosa and the Auerbach's plexus. These are nerve cells that regulate peristalsis, which is a contraction caused by the muscularis externa. And so if we look at that with H&E, this is a muscle on the outside, the muscularis externa, and we're looking at this little box here. And so we can see cross sections of individual smooth muscle cells, and you can see longitudinal views. You can see longitudinal views of these muscle cells, individual muscle cells with the nucleus here, no striations in these. If you stain it with PAS, which periodic acid shift, it stains for high concentrations of carbohydrates. You can see that the basement membrane of smooth muscle cells is PAS positive, like you see in epithelium. And so we do just see the nuclei here, but you see the PS staining around each one of these. If you look at an arteriole, you can see smooth muscle cells. There's a lumen here, these endothelial cells, but we see higher mag of smooth muscle cells. They have these little apical caveoli on the surface that they have there, and you see a high density of fibers, and also you see these fusiform densities, these different densities, and the fibers are actually attaching to these densities to facilitate the contraction of the muscle cell. There's a nucleus, and you can see the high density of actin and myosin in there. In another one, we can see a similar type thing, the densities that are attached to the plasma membrane or inside the cell, and you can see the host of filaments. There are other organelles in through there, but there's a host of actin and myosin filaments in these, and they're not arranged in a certain way in a sarcomere. And so it's called smooth muscle because it's a lack of the periodicity that you see in a striated muscle. Again, we can see some smooth muscle here, and they're being innervated by the nervous system. And here we see the different parts of a nervous system, how they connect, presynaptic, postsynaptic, and a little bit of axon in through there that would have the vesicles inside regulating the contraction of this smooth muscle.
We want to thank the original source of figures, images, and drawings that may have been used here, and they have been identified the sources for which they came in the slides themselves. This is the end of muscle, that is features and functions, types of muscle, smooth muscle, cell characteristics, and locations.